Good morning, everyone. Those who doesn't know me, I'm Kadambod Siddiq. I'm the director of the UWA Institute of Agriculture. And before I start, we must acknowledge that university is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. So on behalf of the University of Western Australia and Institute of Agriculture, I warmly welcome to you this special lecture delivered by Dr. Jun Zou. Jun is a professor of a National Key Laboratory of Crop Genetics Improvement and the College of Plant Science and Technology in Huazhong Agricultural University, Wuhan. Very famous university. We have had collaboration with Huazhong over the decades, I think more than 2 or 3 decades. And the key laboratory is very good. They've got 2 important ones, one on rice and the other one on canola. In fact, it is the hub of canola research and development in China. And we had many visitors here. Jun received her PhD in 2009 from the same university, Huazhong Agricultural University, and worked on research area of Brassica genetics, genomics, and pre-breeding with 62 peer-reviewed research papers, 2 book chapters, 2 patents, and involved in development of 3 hybrid canola cultivars, and mainly focus on creating and exploring Brassica novel genetic diversity by interspecific endogression with genomics-based approaches. She has coordinated more than 15 national and international projects. So Jun is currently visiting UWA as part of the GRDC International Visiting Research Fellowship. This is a prestigious fellowship. GRDC started some decades ago, then it dropped off, and again, the hard work by our own Dr. Shen Cheng, who was instrumental with the support of Wallace and myself to get you here. So we are very pleased and proud to have you visited here. And as part of this visit, Jun also visited Eastern Australia, a number of places, including one more visit to GRDC. So with that, I would like to welcome you on behalf of all of us.
Thank you very much, Professor Stick, for your very, I'm worried for the introduction, and thank you very much for you organizing my visit, and Dr. Shen Chen, Professor Wallace, for you very welcome and organizing my visit here. I'm very, very happy and very, very honored to be here in this very beautiful university to give this presentation. Today my topic is about the account of genome-wide reconstruction of a sort of de novo domestication that was synthetic Brassica napus. Professor Siddiq has just introduced. I come from China, and we are here now, and not so far away. We are in the same time zone, so I think it's very easy to communicate and cooperation with the professors and students here. It's very convenient for our cooperation and academic exchange. And I come from that university. We also have some river around our university. You have very beautiful Swan River, very, very beautiful. I have very nice walking along there. We also have 3 rivers around our university. And I come from that National Key Laboratory of Crop Genetic Improvement in China. I think this maybe this is a very famous or very key laboratory. We are in the 3rd floor for rapeseed. So each floor we have 1 crop from rice, canola, cotton, maize, some crop, mostly on crop genetic improvement. And also Sen graduate from the rice group, very famous Professor Qifazang Zhang, the director of our laboratory of this university. So then we have a rapeseed research center in our university. I think this is also a rapeseed research center in China. Most of the researchers and students graduate from from this university, and we have the different PA different PA focus on different area of genetics, breeding, and genetics, genomics, and breeding. And we also in our university we have from the upstream to the downstream. For example, the genetics, breeding, and also the cultivation, and the economics, and the machinery for the rapeseed industry, and also the plant the nutrition and the soil for rapeseed growth in our university in different college. And USA, we don't have so much land, but it's convenient for comparative for us to can go to the field every day to look at our plants. So that you can see that's the the building for our field station, and that's our different different field trials in our campus. And this is a kind of memory for the founder of the CMS. I think maybe this is the first in practical breeding for the initial for the CMS founded by Professor Fu. He is a Chinese academic, and in the springtime there are a lot of travelers and news reporters. They come to visit our university to see this. So to this introduction to what to our university, our research center, and the rapeseed research in our university. And today my topic is about a kind of annual polyploidization. And as we well know, annual polyploid is very, very important in human life. So not only they are growing as the crop production, and also for evolution and genomics analysis study is very important. So at the beginning, the first we can see in nature, we found and we domesticate and we cultivate the annual polyploids, such as the hexaploid wheat. It's a very big group across the world. And also we can see the cotton, and the napus is also annual polyploid. It's a tetraploid. And from recent years, we can see from the cell paper, there are several groups, research groups, they are domesticated the annual polyploids, the white species, for crop production because of the climate damage and the genetic resource limited, and they also want to create the new genetic cross sources and crop species. So use gene ending and with genomic analysis approach, like for example, Professor Jun Zou, the group domesticated a count of white tetraploid rice to get the But for this de novo domestication, this and a survey review paper from this route, we can also say this is a new direction and has some achievement, and look forward towards the new species. And also the other very practical use is resynthesis or synthetic the annual polyploids, because we know annual polyploids were originated from the diploid or different species. So with that strategy, we can resynthesize with the parent progenitors. We can also synthesize them with the related species because they have some relative very good cross compatibility. So that's because Brassica napus is also a kind of allopolyploid. It's a tetraploid. So this is also very important oilseed crop across the worldwide, especially in Australia. I'm informed last year, this is the second major group now in Australia because of the efforts and from the worldwide and also the Australian people, breeders and genetics here. And in China, Brassica napus also the biggest oilseed crop and also the fifth major crop in China. And majorly they are growing as a oilseed crop, majorly as the oilseed crop. And also some special oil fatty acid components, for example, the high oleic acid. This also we have some cultivars spread from our university as some professor. And also, besides of the oleic production in China recent years, for competition and also for the rotation and also for breed the how to say, help the breeders to give more income for their farming. So for our university, Professor Zhao Yundi, they group introduced some white relative in the same family species to Brassica napus, and I got a count of vegetable Brassica napus with some additional lines from that species, and they get the resistance to very resistance. So it's very healthy, and it's now in the market. We can buy this for vegetable use. And also in our university, also in China, there are a lot of people and company, they grow in the different kind of rapeseed for travel, tourism or like this. And also, yeah, we can say. And then also we breed the cultivars for animal feeding, and also as the the feed for the plants or also for the meal. So we can say the Brassica napus has many. Sorry. It's an important wasted crop, and in production, more than 70% of cultivars was hybrid cultivars. And also, the experience and the evidence from the corn and other crops, the genetic distance between the two parents will affect the heterosis. So we need broad the genetic distance and genetic ratio in Brassica napus. So at the beginning, my professor, when I was a PhD student, he proposed the idea to introduce the genetic variation from a related oilseed crop. From oilseed crop, we can decrease the linkage drag from the vegetable or other this kind of unfavorable traits for oilseed crop. So we will use the different cultivars of Carinata, Rapa, and Jiangsa because they were relatives to Brassica napus. As we know, Brassica napus was originated from Europe, and also they introduced them to China in nearly 1940 to 50s. And then with the local breeding by introducing a lot of local napus, Rapa, because Rapa growing in China as a oilseed crop for 1,000 years. There are a lot of adaptation and genetic diversity. But Brassica napus is a polyploid. They has more advantages to the diploid species. And then for the local breeding, the breeders introducing the local Rapa cultivars to napus to get the new varieties. And also in China and also in India, and they have a lot of genetic diversity for Brassica Jiangsa. So we think this will bring a very good opportunity if we can replace the AC genome of napus with the C, A, or other A from Rapa, Jiangsa, and Carinata. So we can combine a good account of new type Brassica napus. And then when we got this new type Brassica napus, as we know in rice, there are Japanese and Indic. There are two subspecies, and they have a very strong heterosis. So they don't have the species differentiation. They can't hybrid or make crosses, but they have competition, and also we can use their genetic distance and compare between the two parent panel. And so this is the initial is the idea from my professor. And then another moment, I'm PhD students. Then we start with other how to say, the nape, like the nape corticus, yeah. And we work together to then from this to that. So at the beginning, we use several cultivars of carinata and rapa to test if this is true. We can get some heterosis. So we got the rapa to napus, we got rapa carinata to napus. We demonstrate there's heterosis. We grow it in Germany, also in Australia, found in worldwide they present a very strong heterosis. We call it the first generation of new type in napus. And at that moment, from that moment, we cooperate with Professor Colin and Sen, and we have the first cooperation paper in 2010. We evaluated the genetic diversity of our first generation compared with the traditional bean nappers. And then we further screen, use molecular markers and DH microspore culture to improve and homozygote the lines. And then we select lines with more exotic introgrowing and better agronomic and traits and test the heterosis. We select the lines with the high generate combine liability, then cross with more, got the second generation lines. And then we we have other PhD graduate students and worker for more than 600 of in the specific courses with involving hundreds of cultivars of carinata and rapa. Finally, we got 436 different hexploits from ABBCC, these hexploits. And then we bycross with our second generation with the better agronomic performance seed quality traits. Then we got the third generation materials. So from my postdoc and to the annual 5 years, we also work on the improve and selecting the third generation lines of this gene pool. In recent 5 years, we're further introducing Jiangsa to the gene pool. So we call it the fourth generation gene pool. So this is a kind of, we want to integrate the innate genetic variation from 4 species. One from rapa, the local, and also from worldwide rapa. And also from Jiangsa, there's a lot of favorable traits from the A, and also can be introduced from the B genome, and also from the Africa master. And finally, at the moment, we are realized also there are a lot of good trait and genetic diversity in naples. We also need to combine this for high level for the first generation lines. So from the beginning to now, we are doing this root fault to use the same DGMS, that's dominant genetic mass theory, and this then we can get fertile lines, then homozygote them to get inbred line and DH line for breeding. And we also then integrated to the augury mysterilize. This is for hybrid breeding. Then we can get sterilized directly, then we can get it. So from our third generation lines, we cooperate with national and international cooperators, including breeding company and the breeders and the scientists. And we found from from a Chinese company, they screened the resistance lines with long gene resistance. We also screened the materials with very high salt resistance in doing germination, and also the nitrogen using efficiency. And the breeders in our university, they use our materials cross with the male strain lines. We have a series of hybrid cultivars released in Chinese market, in China market. So this is the work in the past nearly 15 or more than 15 years. So in recent years, at the beginning, we know there should be a lot of rich genetic genomic variation because they are introducing from different species. Because of interspecific crosses and hybridization they promote a lot of new variation. And also there are variation from the existing different species, and they are from the, we call it as a pangenome-based variation now from different species. So we know they would be rich genomic variations, but to all the what variation is not we need in breeding. What kind of breeding genomic variation will be fixed and selected later, and how they affect the trait, novel trait, and how they how to say the evolve during intensive selection, and how the genome reconstruct. So we use the genomic-based approach. We further answer these questions. So several years ago, we started to construct the GWAS population by, we got more than 10 years self-pollinated, and we doing a lot of microspore culture, got the homozygote lines with very diverse synthetic lines. We have this GWAS population. We're select some representative lines, might cross with the different ecotype, for example, the semi-winter and winter, the sequenced cultivars of Brassica napus to get the set of potential parent mapping populations. So we're doing several more than 4 years phenotyping work on this GWAS population and the potential population. So in genomics level, we sequence the GWAS population and the mapping population. So this demonstrates some result. This pink circle means our new type in apples with the very advanced and homozygote lines. And that different purple, green, and blue color, they present the semi and the spring and the winter. So I know in Australia, the mostly spring type. So we can say if use new type in apples, still has a lot of genetic difference between the different ecotypes. So in the advanced lines with the sequence data, we can detect a lot of HE, like homologous exchange between. This is not surprising. It's very common in the annual tetraploid. In AACC, napus is especially very common. Sorry. For example, using the Illumine short sequence data, so we can see the coverage increased in one line, and then the coverage decreased in the corresponding homologous chromosome C2. So we further evaluate this homologous exchange between the two chromosomes by the nanopool non-sequence data. So we can say for the non-sequence data, we can say one fragment, they are aligned to the A2, and the other segment, they are aligned to the C2. So we also evaluate this with molecular markers by designing primers. And this large structural variation not only distribute across the whole genome, they're also very stable inherited. Whatever by crossing with the different ecotype in the mapping population, and also for the self-pollinated, they can inherit it very well from generation to generation. That's an example. From different generations, we can detect the B genome integration in these lines. Normally, for the AC exchange, it's very easy, happened in Brassica napus. But for the BC exchange, it's not so easy to happen. We detect the BC exchange in this line. They are across different generation. We can still detect they are very stable inherited for the B integration in this line. And also we found in Brassica napus, this very frequent pseudo employed, not like in wheat. And this phenomenon was found in wheat. And in napus, in the science paper in 2014, they also in synthetic napus by AA and CC, they found this. And we found this is very common in Brassica napus. So in our new type B napus, this pseudo euplate that means for Brassica napus, they have 38 chromosomes. So if you just checked with chromosome counting, you will find this 38 is Brassica napus. But when you use the sequencing and use the fish analysis, you will find it's not a real AACC karyotype. The A2 chromosome has doubled, but the C2 chromosome missed. So because the A2 and the C2 has very high homologous, so they can replace, especially the A2 can be replaced by C2. But this karyotype can very stably inherited in Brassica napus. Even we cross with the semi-vental different ecotype and with the normal karyotype, they are very good chromosome pairing. They are segregating. So we showed they has normal chromosome pairing and segregation in the mapping populations. So, but this will bring very complex molecular marker patterns. When we do genetic mapping with mapping population, not only in Brassica napus, even in other crops, if you doing exotic introducing involving interspecific cross, we think you will bring a lot of new variations in the mapping population. So in normal genotyping analysis, we will ignore the missed and heterozygote markers because we think it's a kind of areas and a kind of missing or like this. But normally they are brought by the genomic structure variation by the parents. So in this mapping populations, we demonstrate how we pick up these ignorant markers by using according to the segregation ratio compared to the parent and in the mapping populations. So we will find the PLV markers and the hemi markers. We select the specific markers. These specific markers, one reason is from they are no compared to the reference genome, they are missing. And also because they are exotic introducing, we don't have in the reference genome. And also because of genomic structure variation. And also because the HE homologous exchange happened, bring this different pattern. So according to this, we screen this across the whole genome level. And then we can identify this kind of markers with very high validation and this marker quantitators. We use them for genetic mapping and QTL mapping later. So we mapped them in the genetic maps. Then we use our phenotypes to do the QTL mapping work. So I just demonstrate one chromosome, for example. For example, normally we just have the green markers if we don't use the PLV and hemi related markers involving the exotic introducing and the structure variation. We were detect a lot of QTLs. But using this, we do genetic mapping, we identify this structure variation related markers in the genetic maps. Then we can identify the loci. Because we can't use the genetic map, we can also have our genome assembly in the letter because this has very complex genomic composition. So by QTL mapping, for example, here we can identify some segment with the RAPA introducing, and we can identify the plant heat related QTL in this region. And also RAPA specific introducing and also cavinata specific introducing across the whole genome. We identified decades of QTLs with this specific introducing in the genetic mapping populations. And in our GWAS populations, use the same strategy. We look the structural variation markers across the GWAS population. So for the normal sleep and in-day markers, we can we identify the hundreds of coattails. But we further use the structural variation markers, we can identify some novel markers that we can't identify, just use these panel markers. And then use this combine the linkage mapping and the GWAS analysis. We identified thousands of coattails involving agronomic traits, yield and yield-related traits, developing flowering time, and this seed quantity traits in our Brassica new type populations. Then we compare it with not published in the conventional bee napus. We will know what is the novel coattail and from the integration or structural variation, what is benefit to the breeding of napus or not. So with based on this coattail analysis, we use some hybrid compilation for further omics data. We cooperate with Rod Snowdown in Germany, Gießen University. We omics sequence hybrid with the window type by our new type in napus cross. So we identified a coattail in F2 and 3 population by coattail mapping and sequence analysis. And in this coattail region, we identified genes. The genes were by genome assembly and analysis. We know they come from the carinata. The C4 to 12, they are carinata genome. By analysis, we know this gene come from carinata. And by genome transcriptome and also the annotation analysis, we know this involving the photosynthesis, and they increase the seed weight. It's not the seed weight for 1,000 seeds, the weight. So we are doing the validation, the gene function validation for these genes. The other example is from the bee genome of carinata. So last slide I presented the example for the C from carinata. And this is from the B of carinata. We found this lines has the B. If we use the normal genetic map, not just like this one, the left side, we saw the special markers involving integration and structuration. We can't identify that gene involving that the novel trait, but use the specific marks. We can identify the nose heel, and by genetic coattail mapping, we can identify there's a very major coattail in this region. And finally, in other combined genomic analysis, we can identify there is a gene integration from the bee genome of carinata, and they change the fatty acid, and then we got the this new type of napus with very no very high 18.1 and 18.2. Like these new materials, they have this bee genome integration, and also by genome annotation, we have found they are most of the gene contribute to the fatty acid metabolism. So this the examples for the trait variation, the per se trait. So for our interesting, we're also more interested on the loci or markers or genome components, they contribute to the heterosis. We want to use materials in hybrid breeding. So we have tested hundreds of hybrids in several years. We use this hybrid for heterosis loci analysis. We identified hundreds of coattails and in the correction pairs. And also with genomic analysis, we can say the structural variation contribute or not to the heterosis. This data we are still in analyzing. So using this testing or this per se trait and the hybrid trait, we want to use them as a test population because we generate new new types each year and each generation. So using the available phenotype and genotypes, maybe we can perform or trying the GS in the new materials. So we cross the different new type genetics with the swing echo type growing in swing in different environments. We got the phenotyping and the genotyping. We tested their performance of genomic prediction. So we cooperate with the APK of Germany, and we're doing the genetic breeding, use different models. In these models, we're considering the conventional markers, the markers with the exotic integration, and the different hydrotic effects. And finally, we found with the integration markers for some trait, the prediction accuracy could be improved. But in this research paper, we didn't show how to plus all of the markers, because at another moment, we just have the GBS genotyping data. But now we will test in whole genome level different markers, how they contribute to the genomic selection models to improve the accuracy. So to summarize my presentation today, firstly, I present how frequent the large-scale structuration drive the genome diversity and the plasticity of the, because this Brassica new types is a kind of new synthetic annual polyploids. And we're doing population-based selection is a kind of very fast, very intensive selection, a kind of not real domesticating with very long history level, but yeah, sort of, yeah, maybe not so accurate. And also we found the structuration were not readily generated, and they can be fixed through influencing on the artificial selection. And we identified hundreds of cotails. We found some cotails novel, and they be contributed to the conventional Brassica area. So we will further validate the genes and the markers. And this also, we are looking for collaborations on this data analysis and further works. And also, in the next step, we need further exploring and improve the breeding value of these synthetic Brassica napus. So my next work, so from the beginning is also kind of this strategy, yeah. And I know there is very good breedings, warnings, and yeah, maybe can also and also Jackie, they has very good the genomics, and can give me and give me suggestions or comments. Then we can improve our group work to further for real breeding, and also improve their work, and also we can think about what kind of since you are interested, we can cooperate further in the coming years. So that's also I come here not just visit, and I also want to express my hospitality and awareness for cooperation, and also welcome you to visit China. So from this, we got the materials, the selection population across years and years. We got the inbred lines and the DH lines. We are still improving and got the new DH population. So by genomics level, we have sequenced hundreds of lines. And this data is under analysis. As I introduced to Jackie and Davey. Maybe we can have a cooperation later for the data analysis or this. And for the screening, for example, the heat resistance and also abatic, I think maybe we can cooperate with the, this kind of traits. And also, I'm very interested in how to put these materials more efficient and faster to breeding. That's what I ask. One is how to use your breeding experience. We can make the real breeding use. I want to, I think this example in Brassica napus, in rice, in cotton, in wheat, all the integration in the specific that has very great contribution to the breeding. We also want in our Brassica area, we can use this as a model to how to improve our breeding and how to will deal this novel variation use genomic-based level. So in my next, I want to do this work and also make them do the breeding, the real breeding. So that's today, and there are some students, a postdoc, not just a professor. So I'm not very sure if my English will introduce very clear for the science or logic inside, but please ask me if you have any questions. Thank you very much. Yeah.
Thank you, Jun. I think we will take some questions because there has been a very interesting topic. A lot of our group has been working in that area. So who want to ask the first question? Wallace.
It's an interesting area. You've generated a lot of diversity over a decade or 2 decades of work, and it's a classical dilemma you face now. You want to make it practically important, but every time you select, you have to lose diversity. So you have this problem of diversity, yes, what do I do with it? I want to make it practical, and I'm going to lose diversity. So how will you protect diversity in this 2 decades long population? Because if you don't protect diversity, you will lose it. Or you can put it in a cold room and store the seeds, and then what? You do nothing. Several years ago, we published a theoretical paper about what to do if you have a diverse population and you want to maintain diversity. It's called evolving gene banks.
Yeah.
And you have to protect that diversity in your gene bank, in the field, when you cross with special techniques such as optimal contribution selection. So I'm just wondering, how will you protect your diversity?
Yeah, that's a very, very good questions. At the beginning, we just want to protect the diversity. So you will see we use this strategy. Cross with this. This is want to protect the diversity. So in strategy level, so we use the population level selection, recurrent population selection. So each year we grow in under a light. They are pollinated by bees. So I often meet the question, what's their parent? Actually, they have very mixed parents. So they are pollinated each other. So they are not only just one line and one line. So they are further pollinated under light. So, and each year we select the materials with good traits, and also they need to have good diversity and this. But even so, yes, the genetic diversity will be still decreased. So each year we also put the good one back to the population, and in population we randomly select and then put the good one inside. That's the one strategy. The other is you can see we also introducing the germ cell and other new things from the outside to inside to our pool. So even so, I think the genetic diversity may be also even decreased. So in recent years, the physically protection is we protect the seeds in the storage, and also we more focus on the anid lines now for breeding. So, I mean, finished gene bank construction level first step. Now we move to the step for how to deem the good ones. We do not, for the first one, if they are cooperators, if they are interested, we can screen, we can go in them, and for the present one, I use the GWAS population and some good lines to cross for breeding. So this is two. I think that we need to separate them. As you know, yes, there will be more and more for breeding And
So my point is, every time you make a cross, unless you are crossing with optimal mating designs, you're going to lose diversity and end up with suboptimal result. Nobody wants to have less than optimum, right? So if in 10 years your population is okay, but it's not really, really good, then everyone would be frustrated. So you have to think about this now, because in 10 years' time you could lose all that diversity and it may be suboptimal.
yeah.
Maybe we can sit together with that paper. This is Wallace's baby now. He's applying
It's
that
a
everywhere.
vertical, you mean we need a vertical selection strategy. Keep the genetic
You could
level.
find yourself fixed for suboptimal characters. If you're fixed, you're now homozygous suboptimal.
Mhm.
You don't want to be there. You don't want to go there by chance. And yet if you have random crossing, you can go by chance to a suboptimal place. You don't want to be there.
Maybe more discussion later on, before you go, because there is some papers done and he has applied that in canola, now going in the bean program and so on. Next question. Anyone else? Yeah.
Have you tested any of your new pipelines for disease resistance?
For what is
For
it?
disease resistance?
For disease
The
resistance.
new lines coming
Yeah,
from
yeah.
this for disease resistance.
Yeah. Unfortunately, we don't. We just grow in the field. We don't have this area now.
Okay.
But there's a lot of beautiful material there.
Yeah.
Yes, please. Yeah.
I was, before all my stuff was on there. I was wondering, you said that you've noticed some large structural variations and whether they've occurred through, and you're assuming most of them have occurred through homologous exchange, so homologous chromosomes. Have you seen any reductions or indications that those homologous exchanges have led to genomic instability? So whether the plants are less fit or they're less viable or they're less fertile. Have you noticed anything like that?
This was a very good question. After selection, at the beginning, their lines has income.
Compatibility.
Yeah, like what the traits. But now these lines, these bad traits has been discarded.
Right.
So I think the HE existed in the present. We can't link to not the phenotype because we have discussed them. But now the HE existed in the at least they are not harmful to other traits. So we identified HE and linked to the traits we screened.
Yeah.
But the trait you mentioned, we can't do. I think we can't do now because not has.
Yeah. I suppose I was wondering, two things. I suppose the first part would be if the ones that are unstable, whatever those combinations are, then you lose the diversity of those, which is just you can't do anything.
Yeah.
But I was wondering, the other thing that I was wondering is if you have these homologous exchanges, can you then, because they've been exchanged, whether it's a sort of reciprocal exchange or something like that, could we see recombination between those subgenomes because of that? Whether there's ongoing recombination between the different subgenomes, because they might have sort of homologous or syntenic regions that would allow them to pair or something like that.
So for pairing, even in chromosome level, for example, in A2 and C2 is no problem for chroming, and even large segment for macros is no problem. But the new variation you mentioned, yes, there still has new variation, especially nearby the areal structuration from the parents. Is that the question?
Yeah, no, I was just wondering whether there was continual interaction between the genomes.
Yeah, I think so.
Yeah.
Like a kind of, they're very boom, and then they will come down, and then they still have some, then come down. But this is a young species in Brassica napus. They very often to have. So this for new type is also even more young, like new baby. So they still, happens, like this.
Can you tell us about the nitrogen use efficiency, because we are now very much interested in mounting a proposal on that. What you just showed that slides,
Yeah.
and what did you find, and is that materials?
Actually, this is cooperated with a professor in our university. He used our very early generation lines to screen the nitrogen efficiency. One arrow has nitrogen and one arrow without hydrogen. Then select the materials. And then they found some materials, then got the DH9s. Then they send back the DH9s to us. And then they do genetics. I think they have found some genes involved in the nitrogen efficiency. We got the lines with the nitrogen efficiency.
So we'll capture all those papers because we need that urgently. As a partner, we are thinking of having Huazhong, or not thinking, we're going to have Huazhong as a partner in that proposal. Any other questions? No? So thank you very much for this wonderful talk and spending time with us. Thank you.
Thank you very much. Thank